So we can look at the second stage of respiration, and that is the Krebs cycle. Sometimes you can call it the citric acid cycle or the tricarboxylic acid cycle. Tri means three, tricarbon, three carbons, and it is a cycle which is moving around and then repeating itself. So it is not moving in a single direction, unlike in glycolysis whereby we're just having a single direction, whereby the 6 carbon atom form the 2 pyruvate molecules, of which something was irreversible. Now in the cycle we see that where we started from we can end there still at the same time. So the cycle, but of course in this cycle we shall have still some ATP being generated, we shall have NAD and some other molecules which are going to see in the nearby future. So we realize that in glycolysis, it is the first stage. It is the first stage of respiration, and then we looked at whatever is being involved, and we are going to see what is involved in the Krebs cycle. So first of all, we can see that by definition, Krebs cycle is the series of enzyme-controlled reactions which occurs in the mitochondrial matrix. So the site for the occurrence of the Krebs cycle is the mitochondrial matrix. Of course, the mitochondria is only going to be found in eukaryotic cells. Eukaryotic cells have membrane-bound organelles. So eukaryotic cells, and in the cytoplasm for prokaryotic cells, remember prokaryotic cells, they don't have a mitochondria, they don't have a nucleus. So for them, they just have no membrane-bound organelles. So their contents are just dispersed within the cell, so we don't have any compartments or membrane-bound organelles. So instead, the Krebs cycle just occur within the cytoplasm. So what is being involved is acetyl-coenzyme A is completely oxidized to carbon dioxide, releasing reduced coenzymes and then ATP, as we are going to see in the steps. So what initiates our Krebs cycle is this. So this is the first initiation of the Krebs cycle. Before we look at the major steps from step number 1 up to step number 8, we look at a pre-stage, or before entering into the major stages, what takes place. The transition stage from the glycolysis stage to the Krebs cycle. So the transition is always occurring from here, whereby we know very well that the end product of the glycolysis was a pyruvate, and remember there were 2 molecules. So it means that if there were 2 molecules, even in these stages, you're going to be having two molecules taking part in the reaction. That's why in each and every molecule we are going to be having here at the end, we are going to be doubling into 2, because this stage is going to be occurring twice. So remember, there are 2 pyruvate molecules which were produced from glycolysis. So the transition stage from glycolysis to Krebs cycle is just, this is a brief stage which is going to be taking place. And how does it take place? We're having a pyruvate molecule, which is 3 carbon atom. 3 carbon atom, remember, 2 pyruvate molecules were formed. Each molecule had 3 carbon atom. So in total, we'll give 6 carbon atoms in the 2 pyruvate molecules. So it is converted to acetyl-coenzyme A. Acetyl-coenzyme A has 2 carbon atoms, of which if there are 2 carbon atoms, it means that decarboxylation occurred from 3 carbon atoms to 2 carbon atoms. Decarboxylation, how did it occur? We're going to see in the equation. So this releases carbon dioxide and then produces reduced NAD. Reduced NAD is the NAD which is having hydrogen. So someone may be wondering, where does acetyl come from? Where does the coenzyme come from? The acetyl comes from the decarboxylation of the pyruvate. So once carbon and then 2 oxygen molecules are lost, two oxygen atoms are lost. Then you're going to just remain with the acetyl group, and that acetyl group is going to combine with the coenzyme A. Coenzyme A, where does it come from? It is already coming from the cell itself. The cell has this component, and what is its relevance? The relevance of coenzyme A is to activate the acetyl group so that it can be highly unstable, hence highly reactive. So at this stage, the reaction, the overall reaction for the transition stage is going to be shown like this. The pyruvate is decarboxylated to release carbon dioxide at that stage. We're going to be having coenzyme A coming from the cell, combines with the acetyl group, form acetyl coenzyme A. But before the acetyl coenzyme A is formed, the NAD, which is oxidized, is reduced to NADH, so which is reduced to NAD. So it means that oxidation of the pyruvate occurs at the same time. So there is oxidation and then decarboxylation taking place at the same time. What is being oxidated? It is the pyruvate being oxidized by the oxidized NAD, and oxidized NAD is reduced to NAD. So it means that this oxidized NAD picks up a hydrogen from the pyruvate to form reduced NAD. That's why we are having NAD as one of the products in the transition stage. So acetyl coenzyme A is formed. The acetyl coenzyme A is a highly reactive molecule due to the presence of the coenzyme A. So we can begin with the step number one. Step number one, that is the condensation reaction. The condensation reaction, of course, we know there is loss of water. Of course, we shall see in the summary of the reaction, there is loss of water. At this stage, you're having the acetyl coenzyme A from the transition stage combining with oxaloacetate, which is a 4-carbon molecule. Acetyl coenzyme is a 2-carbon molecule coming from the transition stage, combined with a 4-carbon molecule, that is oxaloacetate, to form a citrate. In other sources, you will find them calling this citric acid or oxaloacetic acid. So it is all the same. Just because the only difference is that the citrate is an anion of the acid, that is citric acid. Just like you can see sulfuric acid. We can also say that the anion of sulfuric acid is a sulfate. So when I say a sulfate, still it can still remain normal. So if you use citric acid or oxaloacetic acid, it is 100% correct. For example, isocitrate, you can also use isocitric acid. Alpha ketoglutarate, you can also say alpha ketoglutaric acid. Succinate, succinic acid. Fumarate, fumaric acid. It is and it is and it is. So that is the first stage, condensation stage. We're having the acetyl coenzyme A, which is a 2-carbon molecule, combining with oxaloacetate, which is a 4-carbon molecule, forming citric acid or a citrate. So in that case, we are having loss. We are having loss of coenzyme A. Coenzyme A is lost at that stage because in the citrate we don't have coenzyme A. Coenzyme A is already lost, and we still have loss of water molecule. Loss of water at that stage because it is a condensation reaction. So we can go to stage number 2. Stage number 2, it involves isomerization. Isomerization, there is rearrangement of the structure whereby the molecular formula will remain the same, but the structural formula changes. So citric acid or the citrate rearranges to form isocitrate. It rearranges to form the isocitrate. So stage number 3, that is the first decarboxylation. The first decarboxylation means that we are removing a carbon atom, or we are removing carbon at that stage. So decarboxylation. So it means that if we're having the isocitrate, because you know the isocitrate is still having 6 carbon atoms. Remember, we are just having isomerization taking place here. So the citrate was 6 carbon. So it means that if isomerization takes place, the 6 carbons are going to still remain. So in the third stage, you're having first decarboxylation. So we are reducing the number of carbon atoms. We're reducing the number of carbon atoms. In that case, when decarboxylation takes place, we're going to have a loss of carbon dioxide molecule at that stage. And decarboxylation is going to be followed by oxidation. Oxidation at that stage, whereby in case we're having oxidized NAD and reduced NAD. Remember, this oxidized NAD is going to pick up a hydrogen from our isocitrate. So it means that when hydrogen is lost, that is still oxidation. So oxidation occurs at the same time, and decarboxylation occurs at the same time in stage number 3, because we're having a reduced NAD being produced. The reduced NAD, it has taken away hydrogen. It means it has oxidized the isocitrate. As well, we have had a decarboxylation taking place, that is the loss of carbon dioxide. And then what are we going to form? We are going to form alpha-ketoglutarate, or sometimes you can call it alpha-ketogluttaric acid. So in stage number 4, the alpha-ketogluttaric acid, which we had initially, it was 5 carbon molecule. It was a 5 carbon molecule. So we can call it 5 carbon molecule. So again, we are going to have a second successive decarboxylation taking place. So it means that if we had alpha-ketoglutarate, which was having 5 carbon molecules, at this stage when we have the second decarboxylation, it means that the 5 carbon atoms are going to reduce to 4. So alpha-ketoglutarate is going to lose carbon dioxide. So it means the decarboxylation has taken place. Another NADH, reduced NAD, is also formed. It means that still there is successive oxidation. It means that in the alpha-ketoglutarate there is a hydrogen which was removed. So that oxidation takes place to form a succinyl coenzyme A. Now, where did the coenzyme A come from? We said that coenzyme A is already available within the cell. So we don't have that it is formed from this reaction. It is already available. But what is its role? The succinyl alone is highly stable. So it means that it is less reactive. So the coenzyme A combines with the succinyl to make it highly unstable. And the succinyl coenzyme A has 4 carbon atoms. So we enter stage number 5. Stage number 5, we're having substrate level phosphorylation. Substrate level phosphorylation, it means that we are succinyl coenzyme is converted into a succinate. It is converted into a succinate. ATP or GTP is produced directly. So it means that we are having at that stage, we're having a ATP gaining or GTP gaining a phosphate molecule to produce ATP. So we're having GDP guanine diphosphate picking up a phosphate molecule to form it. So energy is released at that stage at substrate level phosphorylation. So in stage number 6, we're having oxidation. Oxidation, the succinate is oxidized to form a fumarate. The fumarate, you can also call it for fumaric acid. So oxidation takes place. What oxidizes at that stage? The oxidizing agent is going to be FAD. So FAD is going to pick up oxygen from the succinate to form a reduced FAD. FAD at that stage is used instead of the NAD. So we are seeing that we are having a reduced FAD formed here at that stage. Remember, as the oxidation is taking place, 2 succinate molecules are oxidized to form 2 fumarate molecules. Why? Because 2 pyruvate molecules were formed from glycolysis. So we are going to have each stage occurring in a double wave. So the 7th stage, we're having what we call hydration. Hydration, we are adding water. So hydration, the fumarate is converted into malate. So it means that here in this stage, we are adding water. So in stage number 8, we're having the final oxidation. The final oxidation. What is, of course, we know the oxidizing agents. We have FAD with the plus, and then NAD with the plus. Those are the common oxidizing agents we are going to be using. So in the final stage, that is oxidation stage, we have the malate being oxidized to form oxaloacetic acid or the oxaloacetate, of which the oxaloacetate is again going to allow the cycle to continue because the cycle is repetitive. Remember, in the initial stages or in the first stage, we had oxaloacetate getting involved. So it means even at the last stage, we must have oxaloacetate being regenerated so that the cycle can continue. So in the second, in part 2, in the second stage, or in part 2 of Krebs cycle, where I'm giving the summary, I'm going to give just the real cycle, of which is going to bring out these real stages. So